Hi, guys. It's been a long time. I really wanted to share this article that Mel put out today. On the post that she made, she says, Wuhan and friends, have you been paying attention to your Epstein borreliosis AIDS? Infinite antigen variation they gave you all for decades. Let's check out the link. Human pathogenic relapsing fever Borrelia found in bats from central China phylogenetically clustered together with relapsing fever Borrelia reported in the New World. This came out in March of 2021. Bats can harbor zoonotic pathogens causing emerging infectious diseases, but their status as host for bacteria is limited. We aim to investigate the distribution, prevalence, and genetic diversity of Borrelia in bats and bat ticks in Hubei province, China, which will give us a better understanding of the risk of Borrelia infection posed by bats and their ticks. During 2018 through 2020, 403 bats were captured from caves in the Hubei province, China. 2 bats were PCR positive for Borrelia. Sequence analysis of RRS, FLAB, and GLPQ genes of positive samples showed 99.55% to 100% similarity to Candidatus Borrelia fani, a novel human pathogenic relapsing fever Borrelia species recently reported in Zambia, Africa, and eastern China, which was clustered together with relapsing fever Borrelia species traditionally reported only in the New World. Multi-locus sequence typing, MLST, and pairwise genetic distances further confirm the Borrelia species in the bats from central China as Candidatus Borrelia fani. No Borrelia DNA was detected in ticks collected from bats. The detection of this human pathogenic relapsing fever Borrelia in bats suggests a wide distribution of this novel relapsing fever Borrelia species in China, which may pose a threat to public health. Bats have a wide range of species and distribution. These mammals have been proposed as the natural reservoirs of pathogens for many years. Previous research focused mainly on viral agents in bats, while infection of Borrelia spirochetes in bats is much neglected. Borrelia species causing Lyme disease and relapsing fever have been found in a wide range of ticks. Relapsing fever has long been recognized as a major cause of disease and death in Africa, but as more and more cases reported in different continents, relapsing fever has become an emergent disease. There is growing evidence that bats can play a role in carrying Borrelia species. In this study, we captured different species of bats from caves during 2018 to 2020 in central China, tested liver tissue for Borrelia by PCR, and applied multi-locus sequence typing to identify this bacterium. Our study found human pathogenic relapsing fever Candidatus Borrelia fani in central China, suggesting that bat-borne Borrelia spirochetes may pose a threat to public health in China and around the world. Relapsing fever is a zoonosis caused by the relapsing fever group spirochetes of the genus Borrelia, and its clinical symptoms include recurrent febrile episodes with headache, myalgia, chills, and nausea. To date, 27 relapsing fever Borrelia species spirochetes have been identified, but additional species have been proposed. Typically, relapsing fever Borrelia were thought to be transmitted by soft ticks, with the exception of the human louse-borne Borrelia recurrentis, as well as the hard tick-borne B. lone starri and B. miyamoto. And I probably pronounced that wrong. Historically, the relapsing fever Borrelia were classified by the one vector, one species concept and in each region. A specific relationship existed between soft ticks and Borrelia. According to the epidemic areas and genetic lineage of the causative agent, the relapsing fever Borrelia were rather arbitrarily divided into 2 subgroups: the Old World Borrelia, the Palearctic and the Afrotropic ecozone, and the New World Borrelia, the Nearctic ecozone. Bats have been have attracted much attention with their close relationship with emerging viral infectious diseases, including Nipah, Hendra, SARS, MERS, and the recent world pandemic COVID-19. It's worth noting that previous research focused mainly on viral agents in bats, while the infection and prevalence of bacteria in bats are much neglected. Knowing whether bats can act as carriers of pathogenic bacteria is a common interest for public health. Ticks, as one of the parasites of bats, act as the most important arthropod vectors for transmitting microbial pathogen to animals and humans. Once bitten by these ticks, risk of infection will increase. Therefore, the role of both bats and their ticks in carrying or transmitting bacteria deserves more study. A recent study found bats and bat soft ticks from a cave in Zambia, Africa, showed a high infection rate with Candidatus Borrelia fani, which was a novel human pathogenic relapsing fever Borrelia species clustered together with the New World relapsing fever Borrelia. It proposed that bats and bat-soft ticks contributed to the environmental cycle of this Borrelia species as host and vectors, respectively. Our group also found that one Myotis bat from Shandong province in eastern China was infected with Candidatus Borrelia fani as the first report of this novel relapsing fever Borrelia species in Asia. Since only a single bat was found to be infected with the Borrelia species, the role of bats in the transmission of Borrelia cannot be determined in China. In this study, we aim to investigate the distribution, prevalence, and genetic diversity of Borrelia in bats and bat ticks from Hubei province, China, which will give us a better understanding of the risk of Borrelia infection posed by bats and their ticks. I'm going to just skip on down and we'll read the discussion, but I hope that you're getting what the implications of this are. And, we have this pandemic going on, and everyone's all afraid of these viruses, but no one is discussing Epstein borreliosis AIDS, infinite antigen variation, spirochetal AIDS, basically. In this study, we found that 2 bats captured from Hubei province of China were positive for Candidatus Borrelia fani, which is recently isolated from a patient diagnosed with relapsing fever in Zambia and clustered together with the relapsing fever Borrelia. Candidatus Borrelia fani is closely related to a group of the New World relapsing fever Borrelia, including the Turicato, or Tecate, I'm not sure how to say that, B. parkeri, and Candidatus Borrelia johnsoni, which were endemic in the USA. Borrelia Turicae and Borrelia parkeri cause tick-borne relapsing fever in humans, and they are predominantly prevalent in the southwestern USA. Candidatus Borrelia johnsoni was first reported in bat soft ticks distributed in the USA. A recent study found that Candidatus Borrelia johnsoni was associated with tick-borne diseases in humans. Previous reports of Borrelia species in bats were only individual cases. A novel Old World relapsing fever Borrelia species, the CPB1, was found responsible for fatal borreliosis in a pipistrelle bat from the UK. Subsequently, Borrelia species CPB1 was detected in soft ticks from bats in France, indicating the association of relapsing fever Borrelia spirochetes and bat soft ticks. A recent study found that bats and bat soft ticks collected from a cave in Zambia showed a high infection rate for Candidatus Borrelia fani, and proposed that bats and bat soft ticks contributed to the environmental cycle of Candidatus Borrelia fani as host and vectors, respectively. However, our previous study found that only 1 out of 145 bats collected from Shandong province, China, was infected with Candidatus Borrelia fani. In this study, only 2 out of 403 bats were positive for Candidatus Borrelia fani. The Borrelia positive bat species found in China in this study include Myotis rickettii in Shandong province, the Rhinolopus pusillus, and Myotis davidi in Hubei province of this study. Myotis rickettii is distributed in China, Vietnam, Laos. Rhinolopus pusillus can be found in southern China, India, Nepal, Myanmar, Vietnam, Thailand, Malaysia, and Indonesia, while Myotis davidi is endemic to China. Besides, the liver and spleen of the two Borrelia positive bats appeared to be enlarged. Borrelia DNA was also detected in spleen, lung, and kidney tissue, as well as blood samples of Borrelia positive bats, indicating multi organ infection resulted from spirochetemia. However, none of the ticks collected from these bats were positive for Borrelia by PCR, possibly due to the small sample size. Given the facts that this relapsing fever Borrelia species infection could be fatal to bats, and the infection rate of this Borrelia species in bats was low, Borrelia infection in bats might be incidental events, just as the case in humans. I venture to argue with that. Further studies are needed to investigate the reservoir and vector of this novel relapsing fever Borrelia species in China. Besides Candidatus Borrelia fani, there were also several reports of the relapsing fever Borrelia found in Africa that phylogenetically clustered together with relapsing fever Borrelia reported in the New World. A new human pathogenic Borrelia species was identified in the Ornithodoros ticks from Tanzania, and it grouped together with the relapsing fever Borrelia reported in the New World rather than the relapsing fever inducing spirochetes that were known to be endemic in East Africa. Another two studies described the discovery of a novel relapsing fever Borrelia candidatus, Borrelia kalaharica, in travelers returning from the Kalahari Desert in South Africa. The presence of Candidatus Borrelia fani in Zambia, as well as the discovery of Borrelia species Lonsdari and Miyamoto, were related to relapsing fever Borrelia, but were transmitted by hard ticks rather than soft ticks, challenged previous taxonomies based largely on microvector specificity and geographic distribution. With more and more studies, we might have a better understanding to the classification of relapsing fever Borrelia. Conclusively, our studies found that the human pathogenic relapsing fever Candidatus Borrelia fani in a new location in China, and further study is needed to determine its true distribution in China. However, with the emergence of this human pathogenic relapsing fever Borrelia species in China, it's important to find the reservoir and vector of it. Well, I will put the link down below to this article, and I just wanted to point out also, war is when you know all vaccines are infected with the real cause, and you continue to do it. Coexistence of ribbon and helical fibrils originating from the human islet amyloid polypeptide 20 through 29. And I will put the link also down below to this. I'd love to hear what your thoughts are on this. Thanks for watching, guys.